Good morning and welcome. Thank you to the organizers for putting this meeting together and to fellow speakers and participants for joining. It's my pleasure to speak with you today. My name is Ian Orozco, and I am one of the scientists at AnaBios Corporation. AnaBios is a contract research organization based here in San Diego. Today I will talk about the advantages of using our human neurons as a translational tool for the development of novel pain therapies. This is a cover story highlighting the impact and prevalence of pain in our society, published by the New Scientist magazine just last year, a couple months after I had started working at AnaBios. This picture illustrates the toll chronic pain can have on an individual. Millions of US adults suffer from chronic pain, about 50 million, 1 in 5. It impinges on the quality of life for the individual and incurs monetary costs in terms of healthcare and lost productivity. Due to the opioid crisis, there's an unmet need to search for novel analgesics, which are minimally addictive. The search for new analgesics starts at the preclinical research level and often entails the use of animal models for pain. The successful treatment for an animal model of pain is quite exciting and is a great milestone, but getting a drug approved on the market is quite challenging to two factors. One is the high cost of funding a clinical trial, which could be in the tens of millions of dollars. And the second challenge is the low success rate, which is often less than 10%. The reasons for the low success rate is not clear, but it's likely that animal models also contribute since they are not predictive of drug effects in humans. There are several examples of this. One is the NK1 receptor antagonist, and the other is the NaV 1.7 sodium channel blockers. In both these cases, these candidates seem to be quite promising in mouse models, but failed to live up to clinical outcomes. The reason for this is not clear, but it's likely that species differences played a role. Species differences could affect the target of interest, how it's expressed in the different species. And the other thing to consider is the mechanism of action, whether it's conserved from rodents to humans, and also possible off-target effects between rodents and humans as well. One way to address the issues of species differences is to perform translational research on human tissue and human cells. This has the potential to validate findings and strategies in the animal mouse models, and then this could be extrapolated to the human case. So this platform has the possibility of minimizing risk heading into clinical trials. AnaBios has a long-standing history and track record of providing high-quality human tissue for translational research. We have a very streamlined process that starts with an organized network with organ procurement offices in hospitals around the country. We have a dedicated staff that's well-trained and employs proprietary techniques and solutions to harvest tissue to minimize ischemia and try to ensure that the tissue is as viable as possible when they arrive at the laboratories at AnaBios. At AnaBios, we process the tissues in various ways. One goal is to fulfill industry contracts. We've worked with several clients in the pain field. This is just a list of the clients that we've worked with in the past. We also perform exploratory research, also with industry as well, in the form of FTE work arrangements. We also do exploratory research with academia. This is a recent publication where a group used our human DRGs to characterize novel NaV 1.7 peptide antagonists. AnaBios has a lot of experience working with the DRGs. It's part of the peripheral nerve fibers. This is the gateway this is the gateway to the pain pathway. The DRGs are quite valuable for studying pain. This system is simple. It involves one principal neuron, which senses noxious stimuli at the periphery. and then the electrical signal is encoded in the neuron, and this gets transmitted to the spinal cord and up to the brain. We've done some exploratory research with the CNS tissue, the spinal cord, and the brain. I will touch on this a little bit later, but a lot of our experience is with the DRGs. The DRGs have electrical activity. It's a quantifiable measure, and the electrical activity correlates with the pain sensation, the increasing pain intensity. The DRGs are quite useful because this tissue is quite enriched. It has a large diversity of nociception targets. For example, there are proteins which are important for pain, such as the voltage-gated NAV 1.8 channel mutations lead to painful neuropathy, and it was exciting to see just 2 months ago Vertex reporting positive results from their phase 2 trials for their NAV 1.8 blocker. It's not only ion channels which are important for pain, but also GPCRs, which could influence the excitability as well. The DRG neurons it's not just DRG neurons themselves. You could also have different cell types in the DRGs, such as the immune cells. When they get stimulated, they could release factors or cytokines, and this could influence the excitability of the DRGs. So the DRG tissue is quite rich in for nociception targets for pain, drug discovery for pain. We hope that our human DRG tissues and cells could help address key steps going from preclinical research to clinical trials. One is to validate the target ID and also the mechanism of action, which was found at the preclinical research level. Our human DRG tissue could also be a source for looking for new targets of pain. We will receive tissue from donors who had experienced extreme pain in their life, such as diabetic neuropathy. So this is a good source for doing gene expression and maybe identify new targets. Our human DRGs, tissue and cells could also be used for characterizing compounds, lead compounds, and optimizing those lead compounds. We could use several metrics, such as efficacy. We could look at how the compounds modulate DRG activity. We could also look at pharmacology metrics such as potency, the IC50, as well as off-target effects. For example, if a compound is doing something to the DRG activity which is unexpected. This is an example of a DRG section. It's been seen with calcitonin G-related peptide. This is a marker for nociception. You can see the soma of the different DRG neurons stained for CGRP. The soma of the DRGs come in, the size is quite heterogeneous. We have different methods for evaluating the quality of our tissue. One is to look at the RIN score, which is a measure of the integrity of the RNA. The scale is from 0 to 10. 9 indicates that the tissue is of a high quality. Our clients want to have high quality tissue because a lot of them do quite sophisticated downstream analysis, such as transcriptomics. This is an example of a recent publication from Genentech where they did transcriptomic analysis from different species, and they had received frozen human DRG from us. We also have our in-house functional assays to evaluate the human DRG neurons. We prepare human DRG cultures, which we analyze. These cultures could be cultured for 2 to 3 weeks, and the neurons will be quite good. These cultures were stained with CGRP, and the distribution of the size of the cells is quite consistent with the size in the sections. So this confirms that the cells that we're looking at in the dish are quite representative of what we see of the in vivo case. The human DRG neurons are electrically active, so we employ the gold standard for looking at electrical signals. This is the manual patch clamp analysis. I myself is trained in electrophysiology. I've been doing it for a while. The last year I've been working with these human DRG cultures almost on a weekly basis, and they're generally quite good. The human DRG neurons are quite large and could be easily identified, and they're not difficult to patch. We've always been able to excite action potentials from them. We have standard protocols looking at excitability. I've been working with clients in an FTE work arrangement, and they will ask for different types of electrophysiology information, biophysical or pharmacology analysis. This is an example of voltage clamp membrane currents where we depolarize the cell and look at the voltage-gated sodium currents. There's different types of NAVs which could contribute to the depolarization. There are some NAVs which are sensitive to the TTX inhibitor. These normally have fast kinetics. And there's also sodium channels which are insensitive to TTX, and they have slower kinetics. Yes, both these NAVs play an important role in generating the action potential. We also look at other ion channels, calcium channels, potassium channels, and TRP channels. Also, the GPCRs also play an important role in pain sensing, the opioid and the mGluRs. And then there are also the neurotransmitter receptors, which are looked at as well. The cells themselves are quiescent. They're growing in media with just growth factors. They don't fire action potentials. However, we could stimulate them with factors such as inflammatory factors, bradykinin or PGE2 in a subset of cells. They will start to induce spontaneous firing. So it induces somewhat of a pseudo pain state for the cell. This is useful for analysis. Our DRGs are also responsive to itch compounds as well. So while the patch clamp analysis provides quite quantitative and biophysical information, it's low throughputs. Another complementary approach we use for functional analysis is calcium imaging. This is useful because many cells at once could be analyzed. For example, up to 150 cells per well. The cells are loaded with the dye, and then they are stimulated in different ways. For example, with pharmacology, we could add capsaicin, which will activate TRPV1. TRPV1 is permeable to calcium. When it's activated, there'll be a rise in intracellular calcium, and then you will see the cells light up. We could also use calcium imaging to indirectly measure voltage-gated sodium activity. We use an electrical field stimulation paradigm. This is a cartoon showing this. A weak stimulation is applied, so we don't activate the high threshold voltage-gated calcium channels, but we do stimulate the sodium channels. And when a large amount of sodium channels are activated, there will be a strong depolarization, and this will in turn activate the calcium channels. There will be an increase in the intracellular calcium levels, and this will activate the fluorescent dye. This is an example in the top panel. This is a cell stimulated with a train of pulses, 10 pulses at 5 Hertz. With each pulse stimulation, there's an increase in the calcium. This calcium step, with 10 pulses, there's 10 calcium steps. We could stop the stimulation, and then the calcium signal will go back down to baseline. During this time, we could apply a drug, in this case, TTX, then we re-stimulate in the same manner. And in this case, the calcium signal is blocked. This means that the voltage-gated sodium channel in this case is sensitive to TTX. We could wash out the first compound, then reapply another compound, for example, lidocaine, which is also NAV blocker, but it blocks all NAV subtypes. So we could use this method to classify other cells. So it's the same protocol, but the other cells are on the dish, and they could have a different readout. For example, in this cell, stimulation combined with TTX does not block the calcium signal. So this suggests that the NAV in this case is resistant to TTX. However, with lidocaine, it will block the calcium signal. So in this bottom cell, it has an NAV which is resistant to TTX. This is probably NAV 1.8, and in this top cell, it's probably it expresses an NAV which is sensitive to TTX. Our DRG cultures are amenable to overexpression and knockdown using viruses. And then after the analysis performed, the DRG cultures could be either fixed or frozen for subsequent protein and RNA characterization. In addition to the electrophysiology and the calcium imaging, we've also started to look at the impact of cancer agents on neurodevelopment of our DRG cultures. We're interested in chemotherapy-induced peripheral neuropathy. These are when patients receiving chemotherapy feel pain, and the incidence is quite variable depending on the drug that's used. We looked at several drugs. One class is the TRK receptor inhibitors, entrectinib and larotrectinib. And patients receiving these drugs will report neuropathy to variable percentages, 18 and 6%. We looked also at oxaliplatin, which is a platinum-based chemotherapy drug. And with oxaliplatin, there's a high incidence report of neuropathy, 74 to 92%. So we wanted to see how these compounds affected our DRG neurons. So they were added to the cultures, starting at DIV0. We added concentrations that are similar to the clinical dose. After 3 days, we looked for changes in morphology, and we quantified neurite length. This is an example of control neurons showing nice neurites after 3 days. When we treated the neurons with entrectinib, we found there was a reduction in the neurite length in a concentration-dependent manner. At the highest concentration, entrectinib reduced the neurite length by more than 50%. When we looked at another TRK receptor inhibitor, larotrectinib, we did not see much of an effect on neurite length. However, when we looked at oxaliplatin, we saw a dramatic reduction in the neurite length. So both entrectinib and oxaliplatin seems to have a neurotoxic effect, reducing neurite outgrowth. It's interesting that in both these cases, they have a higher report of neuropathy, while larotrectinib did not show an impact on neurite length and had the lowest rate of report for neuropathy. So there seems to be some correlation between these drugs impacting neurite length and the report of neuropathy in the clinic. We're still doing further experiments and optimizations. For example, we'd like to add these drugs when the neurons have already established their neurites and see whether they might, these drugs might affect the retraction of the neurites. We hope to get the assay up to a point where it could be useful for the industry to be able to identify compounds which are predictive of neuropathy. Besides the DRG, we're also culturing trigeminal sensory neurons. The trigeminal ganglia is also part of the peripheral nervous system. It senses pain. It innervates the face. This is a trigeminal tissue from a donor. You could see the fibers extending out. We're quite successful at preparing the cultures. These cells could be loaded with calcium dye and will show calcium signals when treated with pharmacology. By patch clamp, they will elicit an action potential. We think this trigeminal sensory neuron prep will be quite useful for clients that are specifically interested in developing compounds for trigeminal neuralgia and migraine. And then moving outside of the peripheral nervous system, we've also begun to do exploratory work on the CNS tissues, the brain and the spinal cord. Both play an important role in pain, so we don't want to neglect them. Recent results found that there was spontaneous activity in the human spinal cord. This is with fMRI. We were interested in this result and wanted to see whether we could see it in our spinal cord slices. This is a spinal cord from a donor. The spinal cord is quite fragile. We could make coronal slices. These are sectioned at 400, 500 micrometers. To evaluate the spinal cord slices, we add a dye, a TTC, and this will stain the viable tissue. And then this is a nice healthy tissue. You could see the staining in the dorsal and the ventral horn. And those, if it's a good prep, then we will culture these spinal cord slices. Our spinal cord tissue and slices have a high RIN score, which is indicative of high quality tissue. We've also done analysis to look at the neuroanatomy. This is a collaborative immunostaining with Dr. Ayaderola at the NIH. He stained the slices for CGRP. This is a marker in DRGs, a nociceptive marker. It's on the DRG afferents, and this is the staining here is consistent, looks correct. It's in the dorsal horn. So this suggests that the neuroanatomy of our spinal cord slices is preserved compared to the in vivo case. Lastly, when we have good spinal cord slices, we will load a calcium dye and look at transients. This is a sped up movie showing the calcium transients. There's a lot in this one. The quantification is shown on the right with a raster plot. You could see the spontaneous activity is quite persists for at least half an hour, which is good for performing pharmacology. These experiments are quite difficult to do. It requires very good spinal cord slices, and you have to make sure that the transients don't run down. We've been able to do some pharmacology, and we found that the calcium transits do depend on action potential generation when we add NAV blockers, and also are sensitive when we add blockers for synaptic transmission. Lastly, we've also started to recover human brain. This organ is quite fragile as well, like the spinal cord. The RIN scores also could be quite high, and suggesting that the tissue would be also could be quite good for gene expression analysis. We've also done characterization of the neuroanatomy using H&E staining. Lastly, I've come to the final slide. To recap, AnaBios is a contract research organization. We provide high quality ex vivo tissue for translational research. A lot of our experiences with the TRGs, they're a useful tissue for drug development. We've also done exploratory work on the CNS tissue, the spinal cord and the brain, but this is the early stages. AnaBios also prepares organ tissue from other regions. We perform action potential measurements and contractions with heart and primary cardiomyocytes. We prepare lung slices as well and primary hepatocytes from the liver. So the goal of AnaBios is to work with clients to validate therapeutic approaches, and this will hopefully minimize risk going into clinical trials. With our platform, AnaBios hopes to tilt the balance so that we could get more pain drugs approved for the market. Lastly, I'd like to thank our leadership team, both Andre Getty, the CEO, and also Kevin Carlin, the Vice President of our Neuroscience Department, are here today.